Hi, everybody. Welcome to Lab 5. This is our enzyme lab. So hopefully you've had a chance to watch the lecture on enzymes. Hopefully you've had a chance to read through the lab and maybe answer some of the questions at the beginning. But essentially, enzymes are just proteins. And if you remember, proteins are polypeptides. So they're a polymer of amino acids. And the order and sequence of those amino acids and their characteristics based on each one's functional group dictates the shape and also the function of that enzyme. And so we have two different classes of enzymes. We have enzymes that can build things up, like that repair tissue and things like that. And then we also have enzymes that break things down that are catabolic. So we've got anabolic and catabolic enzymes. Today, the peroxisome is what we're going to be looking at. I'm going to skip through a little bit of this stuff. So we talked about the catabolic enzymes as being exergonic, as energy releasing. We talked about endergonic, are the enzymes that are anabolic, or endergonic, meaning they require the input of energy from somewhere else to operate. And today we're going to look at the enzyme peroxidase in the cell. And so the question that we're going to be answering today is, so what sort of variables will affect the breakdown of hydrogen peroxide by the peroxidase enzymes that are in each piece of these living organisms? So you should have a potato, you should have an apple, you should have, he says raw meat. I don't have any raw meat. I don't eat a lot of meat, so I apologize for that. But you guys can definitely, if you have hamburger or raw chicken, you can substitute that. I just picked a pomegranate off our pomegranate tree, and then I picked some leaves off of one of the Madagascar primrose that we have out back. And so we'll test that too. So those are my two options. So you need four things that you're going to test. You need four tissues in total. So the peroxisome inside of a eukaryotic cell is an enzyme that breaks down both fatty acids and alcohol. So the peroxisome breaks down molecules by oxidizing them, and it transfers hydrogen atoms to oxygen. That produces hydrogen peroxide as a byproduct, which is harmful to cells. So by breaking things down inside the cell, the peroxisome, that organelle, is creating byproducts that are toxic. And that byproduct in this case is hydrogen peroxide. And so the cells have an enzyme that breaks down hydrogen peroxide, and those are called peroxidase enzymes. And so we have the balanced equation on the next page at the top of page 3, which says, okay, if we have two hydrogen peroxide molecules plus a peroxidase enzyme, what the peroxidase will do is it will chop off part of the oxygen and release that as a gas, and then it will also leave behind some water. So that's how the peroxidase works. Now, we have to talk about what substrate, enzyme, and products are in a reaction. So a substrate is just anything that the enzyme is working on. The substrate, once it's worked on by the enzyme, will become the product. And so you can fill in those blanks, right? Our substrate in this case is going to be the hydrogen peroxide. Our enzyme is peroxidase, and then our product is going to be the H2O and water. All right, yeah, H2O and oxygen, my bad. Is oxygen released from the reactions a gas? And the answer is yes, O2 is a gas. Is the above reaction endergonic or exergonic? Ooh, that's a good question. Is it breaking something down or is it building something up? And I would say that it's breaking something down. So you can circle the right one for that. Based on the figure below, what observable products of this reaction would you use to determine if the tissue contains the enzyme peroxidase? Well, for this one, obviously the bubbles of oxygen gas that are the byproduct, or the product, if you will, of the enzymatic reaction are going to be what we test for. And so now we get to do our laboratory. So a couple of things that you guys are going to need. You're going to need a knife, like a small paring knife or a regular kitchen knife. You're going to need your scale so we can measure out the proper amount in grams of each one of our organic materials. You're going to need 4 test tubes. Now the problem, the test tubes are great that we got the test tubes. The problem is that we don't have a test tube rack, like a rack to hold them in. But I just took like a small mason jar, any small container that is completely transparent will work. So you just need something that you can see through, so you can count and see if count the bubbles, right? Okay, so let's get started. We're gonna need to take 10 milliliters of our hydrogen peroxide. So you're gonna need to get your hydrogen peroxide out of your kit. You're gonna need your graduated cylinder. And then for the rest of our lab, we're gonna need some vinegar as an acid, and then we're gonna need some baking soda as a base. You'll also need ice cubes and hot water, but you can get hot water from your tap and you can get ice cubes from your freezer. So I don't have those out right now. Okay, so what we need to do is we need to measure out 10 mils of hydrogen peroxide and add it to each of those 4 test tubes. Now I'm gonna pause the video and just let you guys do that. You guys go ahead and pause the video too. Okay, so should have 4 tubes, each with 10 milliliters of 3% hydrogen peroxide. Now what you could do, what I did was I used a transfer pipette and I poured the bottle. So I poured the bottle until it was almost 10, and then I used a transfer pipette to put the rest in. So anyways, so we got our four test tubes. What we need to do next is get our living tissues and then write in the tissue that you're choosing on table 1 below. So that means we have to flip the page, and it looks like on page 4, although it's not labeled, page 4, we're going to write in our tissue 3 and our tissue 4. After you've written those in, what we're going to do is cut and weigh out 2 grams of each of the 4 tissues. What I recommend is chopping up the tissue first so it's ready to go, and then having a little weigh boat. You can even take the lid to your scale and place it face up so it's not covering it. And then you can tear your scale so it reads 0, and then add your chopped up tissue to the scale until it reads 2 grams. And then once you have those 2 grams, you can set it off to the side. And then that way, when we add all of the tissues to the tubes, we do it all at the same time. So we can take our measurements at 1 minute, 3 minutes, and 5 minutes. So I'm gonna let you guys go ahead and start chopping those up. I'm gonna chop my stuff up, and then I'm pausing the video while I do that, and then we'll do the rest together. All right. So what I did was I weighed out 2 grams of apple, 2 grams of potato, 2 grams of pomegranate, and 2 grams of leaves. Now I need to dice these up really small so that the hydrogen peroxide is able to cover as much surface area as it can. And so I'm going to do that right now. I'm going to pause the video one more time, chop them up real good, and then we'll add them to our test tubes. Okay, so I've got all of my stuff chopped up. We're about ready to add it to our test tubes. So one thing we got to keep in mind is that I'm not going to be able to add them all at exactly the same time. So what I want to do is keep track of which one I started with and start my timer right when I add it to that one. And then so I'll start my timer when I add it to it, and then I'll add each one to the next tubes and so on. And then at 1 minute, I'm going to read the first one, and then I'm going to read the second one, then I'll read the third one, and then the fourth one. And so hopefully that'll be about a minute each. And so that's how we're going to do it. Make sure that you have your phone so that you can set a timer or stopwatch to record every minute, 3 minutes, and 5 minutes. Yeah. Okay, let's begin. So I got my phone out. It's ready to go. I got my stopwatch set up. I'm going to start with the apple first. So I'm going to grab my apple. I'm going to put it in there. And then it's not touching the hydrogen peroxide yet. It's just kind of sitting at the top. And then I'm going to knock it down and start it. Okay, so apple is started. I'll even use my transfer pipette to kind of push it down. Okay. So there's apple. And then I'm looking at my clock right now, and I'm seeing 13, 14, 15. So I'm adding the potato now. So at about 15 seconds, that's when the potato is going to go. Holy cow. I would say the potato has a lot of cheese. The thing's foamy as hell. Okay, 30 seconds. So I'm going to add my pomegranate. So I'm adding that at about 30 seconds. Okay. And then I'm adding my chopped up marigold, which was a little bit of a pain in the ass. I don't know if I would recommend using leaves. They just kind of like, there's just so much of them, you know? To make 2 grams. So this one might be I might have tanked this one. Maybe use an onion. Okay, and then we're at a minute now, so I'm gonna read my first one, which was the apple. I would say like that's a 1. My potato is like a 5 for sure. Pomegranate's like a 1. And then this one, yeah, I'd give that a 0. That doesn't look like it's doing anything. I forgot to get a pen, so I better hurry. All right. So my potato was a 5 at 1 minute. My apple was a 1. My pomegranate was a 1, and then my leaf was like a 0. All right. So then we just wait for another minute, and it'll be 3 minutes and just a minute, and then I'll record them again. My potato's like gonna overflow. See the potato? It's just like. Apple, got my pomegranate, and our leaf. Oh yeah, the leaf's starting to make some. All right, we're getting to our 3-minute mark. My potato's about to overflow. All right, we're at 3 minutes. So the first one I'm gonna look at is my apple. Yeah, I'll give that a looks more. So I'll say like a 2. My potato is next. I'd say that's a 5 still. And pomegranate, I'll give that a 2. And then my leaf, I'd say the leaf's at a 2 now too. All right, my potato's like erupting out of the jar. So now we got another minute, just chilling, and then we will rank the peroxidase activity in the tissues from highest to lowest. And then don't forget to take a picture of this after you're done recording the data. So we got about 1 more minute, and then we'll record the data, and then we'll switch over to hot and cold. So the hot and cold will need hot water, we'll need cold water, and I think that's it. Yeah. So a good question for this would be, what type of scientific investigation is this? Would this be descriptive, comparative, or experimental? Well, I'm taking one variable and comparing it across different groups, so that would be comparative. I'm comparing the peroxidase activity between many different tissues. What would be my dependent variable? That's a good question. What's my dependent variable? We're approaching 5 minutes. The dependent variable is going to be the amount of bubbles produced. All right, I'm at 5 minutes now, so I'm going to check my apple. Yeah, still cruising along at a 2. My potato is definitely a 5. It's like out of control. And then I got my pomegranate. I'd say the pomegranate dropped down to a 1. Now let's check out this leaf. think the problem with the leaf is that it's not sticking into the peroxide. And I'd say the leaf probably went down to a 1 as well. Okay, so that experiment's done. And so, well, let's figure this out. Let's rank them from highest to lowest. So for me, my potato is definitely 1. My apple was definitely 2. My pomegranate was 3, and my leaf was 4. Okay, and so what I would do right now is I would take a photo of me, cheese, and I would submit that for my, well, I'd save it on my phone, and then I'd submit it for my assignment later. So the next question we have is, what is the effect of temperature on peroxidase? And so we're going to fill up a beaker. We're going to need our beakers from our lab kit. So we're going to need 1 beaker with some ice in it. We're going to need another beaker with hot water. And yep. And I will be right back. Okay. So for this next one, we're going to need 3 test tubes, 2 beakers. 1 beaker is going to have hot water in it. 1 beaker is going to have ice water in it. And then the other test tube is going to have just be at room temperature. And so what you can do right now is you can take these test tubes and clean them out, and then we'll get started. Also, before we do that, also remember, it says to take one of the tissues that, kind of mildly reacted. And so what I'm going to do is I'm going to take my apple, and I'm going to put the apple in the tube for mine. So I'm going to cut up some more apple. Plus, I got a lot of apple. All right, so first things first, we need to add 10 mils of hydrogen peroxide to each one of these tubes. Yeah, so we're going to do that first. So let's take 10 mils, add it to each one of these tubes. Now, what I did for my hot water is I took my beaker and I put like hot tap water in it, and then I stuck it in the microwave. And then hopefully that, I don't want to use that one. And so I just microwaved it. If it melts my beaker, at least I can tell you not to do that. All right. So while that's getting nice and hot, I'm going to pour 10 mils in that one. There's my hot water. 10 mils in that one. And then one more 10 mils. Let's do 10 more mils. Actually, I'll tell you what. All right. So this water is really hot. Since we have to let these soak for 5 minutes, I'm just going to take the 2 I filled up and I'm going to put them in here. Hopefully they don't fall over. We're going to have a mess. Okay, perfect. Okay. So we're going to let those sit for 5 minutes. I'm going to set a timer. While those are sitting for 5 minutes, I'm going to fill up this other one. Okay. Timer 5 minutes. Yeah, and then we'll fill up our last test tube with 10 mils of hydrogen peroxide. So again, just pouring. Got to get the bottom of the meniscus to the 10 mil line. Perfect. And then this will be my room temperature one. Also, while those are heating and cooling respectively, what we can also do is we can take our apple, which is what I'm going to do. I'm going to do the apple, and I'm going to go ahead and weigh out 6 grams. Now, I'm not going to weigh it out all at once. I'm just going to do 2-gram chunks. So as soon as it's zeroed, we'll put a little bit on there. That's 3.4, 2.3. Oops. 2.1, 1.9. Come on now. Come on. All right, so 2 grams of that chunk. I'll set that right there. 2 grams of the next chunk. I was like, that's way too big. I cut that right in half perfectly. There we go. So there's another 2 grams. That one's almost perfect. There we go. All right, so now I got my 3 chunks of apple. I still got 3 minutes to wait until the water's hot and cold, respectively. But what I can do, set my scale off to the side. I can take this apple that I chopped up here, and I can wipe down the counter a little bit so I'm not adding any chunks of plant leaf to my apple. Yeah. And I can chop up each one of these apples really nicely. So you guys can do that too while you're following along. So we'll chop up our apple really good. All right, and then after this, we've got one more test to do. And that test is going to be on acid and bases. So we're going to see how pH affects the activity of an enzyme. So you have to consider that enzymes are designed by nature to operate at a particular temperature and a particular pH. And the reason for that is a lot of animals have homeostasis. So like for endotherms like us, warm-blooded animals, we tend to regulate our body temperature at a particular temperature. And you might wonder why. Like, why is 98.6 our ideal temperature for us? And the answer is because our enzymes that are coded for by our genes are designed by nature, by evolution, by our natural selection. Our enzymes are designed to operate most efficiently, most effectively at a certain temperature. And so that's why we're going to test the effect of pH and temperature. So what you can imagine is that it's possible that the temperature is going to negatively affect on either end of the spectrum. If you make an enzyme too cold, maybe it's too stiff and it can't move around the substrate. If you heat it up too hot, maybe the protein's too flexible and bendy and it can't lock onto the substrate. Maybe room temperature will be closest to body temperature for the peroxidase enzymes. So we'll see what happens. We got about a minute left. I'm just going to pause the video. I'll unpause it when we're ready. All right, so now we're ready to add our substrate, if you will, to our enzyme, I should say. The enzyme is in the fruit. The substrate is the hydrogen peroxide. So what I'm going to do is I'm going to get my stopwatch ready. And in the lab manual, it says to take them out of the cold water and out of the hot water. But I feel like I'm just gonna leave mine in there. Like it's not hurting anything, and it'll hold and stabilize the temperature. So I'm just gonna leave them in there. I'm gonna add my apple to the cold first, and then I'll add it to the hot, and then I'll add it to the room temperature one. And so I'm gonna take out my cold tube. I'm gonna take my chunks of apple, add them in, 2 grams worth. And it hasn't touched the peroxide yet. So I'm just going to go start. There we go. I'm going to put it back in the cold. Now hopefully I can do these other ones quickly so I can read that first one after a minute. So I'm going to do this fast. I'm going to stuff this in there. Put it in the hot water one. This one's like really hot. It's like burning my hand. All right, there we go. Hot water. Shake it. Put it down in the hot water. And then finally I got 30 seconds to add this last one at room temp before my first reading. 20 seconds. There we go. All right. There we go. This one's at room temp. All right, so let's get ready. I got my pen. Record the bubbles at 1, 3, and 5 minutes. We're approaching 1 minute. I'm going to check the cold first. 55, 56, 57, 58, 59. Let's see what we got. Huh. I see like 0 bubbles in the cold. Okay. Let's check the hot. Mm, I see like 1. I see some, so I'm gonna put 1. Let's put that back in there. Then let's check the room temperature one. I'd say there's 2 in that one. So a little bit more in the hot, just a little bit. And then at 3 minutes, we'll check it again. So I'm just gonna pause the video. Okay, we're approaching the 3-minute mark. Let's check our cold. Okay, I see some bubbles in the cold. I'm gonna put a 1 in the cold. See how the hot's doing. I give the hot a 1 again. And then the room temperature one is definitely better. I would say it's probably like a 2. It's definitely got more than the hot, so I'm gonna put a 2. All right, now we just have to wait 1 more minute, and then we'll take our final measurement. All right, we are at 5 minutes. So let's check the cold one first. I see some bubbles. I'm gonna give that a 1. Try the other one. Hot. Hot's got the same as the cold, so I'm gonna give it a 1. Let's try this other one. Yeah, the room temperature is definitely more too. Not by much, but it's definitely more. All right, and so that's it. We used apple. I'm gonna write my name of my tissue in there. And then we can rank the peroxidase activity based on temperature. And so I would say room was highest, then hot, then cold. There you go. All right, so we're gonna get ready for the next one. So getting ready for the next one, we're gonna do pH. So we gotta clear out our test tubes. Let's go ahead and rinse these out. And then we're gonna fill them back up with 10 mils of our hydrogen peroxide. All right, in this one, we're gonna have 3 test tubes. We're gonna test the effective pH. So each of these is gonna have 10 mils of hydrogen peroxide. One of them is gonna have vinegar in it. The other one's gonna have baking soda in it. And then finally, the last one's just gonna be regular. So it said to use a tissue from section 1 that had, an average amount of bubbles, which I would say would be our apple. So we're gonna use our apple again. We're gonna cut up, just like we did the first time, we're gonna cut up 2 grams and put it in each tube. But first we need to put 10 milliliters of our hydrogen peroxide in each tube. So go ahead and do that. I'm gonna pause the video. You guys can do that too. Okay, so here we go. I've got our 3 test tubes for the pH experiment, each with 10 mils of hydrogen peroxide. And now in the instructions, it says to add 3 drops of either vinegar or lemon juice. So I'm going to take my transfer pipette. I got to make sure you keep track of which one's which. I'm going to do the far left one is acid. 1, 2, 3. Okay. And then what you can do, he says, you can take your thumb, if your thumb's big enough, and just kind of mix it up, homogenize it. Okay. The next one is baking soda. He says to use 1/4 gram, so that's 0.25 grams. So all I'm going to do is just take the blade edge of my knife, stick it into my baking soda, take out just a little tiny bit. Yeah, that's 0.6. Take away some of that. If a cop walked in my house right now, I'd be like, "What are you doing?" What is that white powder, sir? It's baking soda, I swear. Yeah. 0.4. 0.3. That's close enough. So I'm going to go with 0.3. We're going to add this to the other test tube. It's like, what are you cooking here, buddy? All right. So there's our 0.3. That's our basic solution. And then we got our normal one. Okay. Acid, base, regular. Now we need to cut up three 2-gram sections of apple. Make sure I get this baking soda off here. Right, baking soda. Okay. So we're going to cut up 2 grams of apple. Not bad, 2.3. 2 grams there. Two grams there. Oh, almost. 2 grams there. It's making like 3 little piles of apple. And then we'll chop those up really good. Two grams there. Okay, so go ahead and chop these up real good. I'm going to pause the video and then we'll add them to our containers. All right, same thing as before. So we'll start with the acid, then I'll start my timer, and then we'll take our first measurement at a minute. So I'm going to take my first little bit of apple. 2 grams of apple, shove it in the acid, and then hit start, shake it up, and then hopefully we can get these other ones knocked out before we get to our minute reading. So I'm going to hustle. Let's get the apple added in. And as long as it's close to 2 grams, it's not, I mean, it's not to be perfect. You don't have to get every little chunk in there. Okay, so there's my base. I'm at 23 seconds. So there's our base. And then here's our neutral pH. If hydrogen peroxide is even neutral, I don't know if it is. All right, we got 20 seconds before our first reading. Okay. All right, and shake it up. All right, and there's our neutral. So now I'm almost at a minute. Let's get ready to take our first measurements. First one's gonna be acid. 1 minute. Oh, it's pretty legit. I'm gonna say 2. Let's check the base. This one I'm gonna give 3. This one actually looks not quite at a minute yet. I might even give that a 4. Okay. And finally the neutral one. Oh yeah, this one's zipping along too. I would give this one, gosh, yeah, 4. Maybe even a 5. It's crazy. All right, so then we got a minute to wait until we get to our 3-minute mark. We're just hoping that these tissues can stay down. And the problem is the apple floats, so it's not like all the way at the bottom. So you're not seeing the chunks rise up, or you're not seeing the bubbles go away from the apple because the apple's sitting at the top. All right, so we got about 45 seconds. I'm just gonna pause it. Okay, we're almost at 3 minutes. The first one we're gonna check is our acid. I'm gonna give that another 2. Let's check our base. That one seems like it's dropped a little bit. So I'm gonna give that a 3. And then our neutral one. This one looks like it dropped a little bit too. So I'm gonna give it I'm gonna give this one a 3 as well. Then we got 2 more minutes, so I'm just gonna, make sure that the hydrogen peroxide that's in contact with the surface of the apple is fresh. And let them sit for another 2 minutes, and we'll take our next measurements. I'm gonna pause it again. All right. So even if yours aren't reacting exactly like mine, that's fine too. You might have used a different apple. Mine were Envy apples I got at Costco. So we're at 5 minutes now. I'm looking at the acid. I'm gonna give that one like a 1. I'm gonna look at the base. Give that a 2. I'm gonna look at the neutral one. It's probably like a 2 as well. All right, and there you have it. Okay, so that's about it for the walkthrough with the experiment, but now we can answer some of these questions together. So I'm gonna turn off my phone. All right, so let's see, tissue used, I used apple, let's see. Rank the peroxidase activity based on pH. I'd say the neutral is highest, then base, and then acid. But the base and the base and neutral are pretty close. And then take another photo of you and your experimental setup. Yeah. Okay. Now let's go to post-lab questions, and I want to help you guys with these, so let's work through them together. So how do we know a reaction was occurring when we added different tissues to the hydrogen peroxide? Well, what was the product that was produced from the tissues? What was the dependent variable? What was it that we were measuring? Bubbles. So as the oxygen gas was produced. All right, since living tissues are able to break down hydrogen peroxide, does this suggest that living organisms are normally exposed to hydrogen peroxide? Objection, leading the witness. Of course. Like, why would you have an enzyme that can break down hydrogen peroxide if you never get exposed to it? So I would say yes, of course that makes sense. All right, what is the general function of an enzyme? It's either building or breaking down. Building or breaking down organic matter. Write the specific reaction we examined today like a chemical reaction. Label the substrate, enzyme, and product. Okay, so what was it? It was H2O2 plus, what did we say? What did we call this? Tissue. H2O2 plus tissue plus enzyme. Oh, wait, the enzyme was the tissue. My bad. Ugh. H2O2 plus tissue goes to be 2H2O plus O2 plus tissue. And then you can label the substrate was the hydrogen peroxide, right? The tissue was the enzyme. The products was the O2 gas and the water. And then the tissue was the enzyme. So the enzyme was unchanged by the reaction. All right, what is the function of an active site of an enzyme? You may need to look this up. But I'm going to save you some time because this lab is long. So let's do it. The active site is the site that targets the substrate. So it's like, let's see. So the active site would be what fits the substrate. So it's what recognizes the substrate. So the active site is the portion of the enzyme that reacts with the substrate. And what is the suffix on most enzyme names? Well, what is the enzyme we were working with? Peroxidase, ASE. So it's a dash ASE. What is the role of the organelle called the peroxisome? Well, didn't we say that earlier? So we go back to the beginning of the lab. The first page beginning. And it's an organelle in the cell that breaks down fatty acids and alcohol. There you go. So it breaks down fatty acids and alcohol. And fatty acids are what type of macromolecule? A lipid. Is it the monomer or polymer form? It's the monomer form. Okay, list how the amount of each substance changed as the peroxidase reaction moves towards completion by filling in the table below. All right, so the substrate, as the reaction was going on and the enzyme was reacting with hydrogen peroxide, it was breaking down hydrogen peroxide. So it was removing the molecule hydrogen peroxide from the solution. So that would be a decrease for that one. And the peroxidase enzyme. So enzymes are unchanged by reaction, so that's probably no change. And then the product would be what the hydrogen peroxide is being converted to. So the product was water and oxygen. So as the reaction proceeds, we'd expect to see more and more water and carbon dioxide. So that would be an increase. And there you have it, folks. That, I believe, was it for the lab. And so you have your instructions for submission. It said you can either print off this lab and fill it out, right? And then you'll use your PDF Maker app on your phone, or you can take photos of each page. You can upload it as a PDF. JPEGs work just fine. I'm totally fine with PDFs or JPEGs. Like if you can do both, if you can do one or the other, I'm happy. Or you can make a digital copy, so you can upload this, or you can make a copy of this in your own Google Documents and edit it straight from the Google Doc, and then you don't have to deal with paper and stuff getting spilled on it like I did. And then you can save that digital copy as a PDF, and then you can upload that with those 3 photos you took of you and your experiment to Canvas, and that is it. I hope you guys had a good time. This was kind of fun. All right, I'll see you later.